Hello everyone, my name is Karan Kapil, and today in this presentation we are going to discuss two very interesting and fascinating topic of genetics, that is color blindness and hemophilia. There is a reason why I am discussing both these topics under a common slide, as it is very usual that color blindness and hemophilia both are X-linked recessive disorders. That means the gene responsible for the formation of the disease color blindness as well as hemophilia is present on chromosome X, and the mutation which result in onset of disease is a recessive mutation. So let's start our discussion with the topic color blindness, which is also known as color vision deficiency. So there is two type of color vision deficiency, or you can say color blindness, that is red green color vision deficiency, and blue color vision deficiency, or you can say red green color blindness and blue color blindness. So the first type, red green color blindness, is a very common type. In this, the affected individual have difficulty in distinguishing between some shades of red, green, and yellow, whereas the blue color blindness is quite rare disorder, and then the affected individual will face difficulty in differentiating shades of blue and green. I will like to break a myth that many of the people believe that the person who is color blind will not be able to see the distinct color. Like if I am green, red or green color blind, then I am not able to see the red color, or you can say the green color. And if I am having a blue color vision deficiency, then like I will not be able to see the blue color. No, this is not true. The main difficulty the person who is having color blindness always face, that the person will not be, the patient particularly who is having the disease color blindness, will not be able to distinguish between different shades of the color, and will also face it very difficult to pick out the color from the bunch of colors. Like if I having different color pebbles and I am having a red color blindness, then I will feel I will um, I will be having difficulty in picking up the red pebble out of the bunch of the pebbles. So in general, if describe color blindness, then the problem arise due to, you can say, deficiency or total absence of color sensitive pigments present in corn cells. So as we know, corn cells are a special type of nerve cells present in the innermost layer of the eye known as retina, along with the rod cells also. Okay. So for a general knowledge, the rod cells are responsible for low light vision, whereas the cone cells are responsible for color vision along with the bright light vision. So the cone cell, on the basis of which wavelength they are picking up, can be divided into three subtypes like S cones, M cones, and L cones. The first word signify which wavelength they are picking. Like the S cone is sensitive to the shorter wavelengths of light. The M cone will be sensitive to the medium wavelength of light, and the L cone will be sensitive to the long wavelength of light. Like I here colored different cone cells on the basis of the color which they are going to pick up. Like S cone, which is sensitive to the short wavelength of light, is responsible for the blue color vision. The M cone, which is picking the medium wavelength of light, is responsible for green color vision, and the L cone, which is picking the long wavelength of light, is responsible for the red color vision. Okay, so if any of these pigment or every pigment is absent, then it will result in the onset of a disease color blindness. So here is a basic flow chart about the cone cells, where S cones, M cones, and L cones are present. In S cone, there is particularly, you can say, in the S cone cells, there is a protein present inside the cells which is responsible for the blue color light, because S is picking up the lower wavelengths of light, or you can say shorter wavelengths of light. So the protein which is present inside these cells are coded by a special protein. Sorry, is it coded by a special gene known as opsinin 1 SW. ठीक है, SW is for the shorter wavelength, and the gene is present on chromosomal number 7. It is autosomal, you can say, gene, not a sex chromosomal related gene. Now the M cone, the medium wavelength cone, the protein present inside these cells are coded by the gene opsinin 1 MW, that medium wavelength, and that gene is particularly present on the X chromosome. And then the alcone, which is responsible for the longer wavelength, or you can say red color, are coded by a gene that is opsinin 1 LW, longer wavelength gene, and that gene is also present on X chromosome. So now you will be having a framework that why red and green color blindness are correlated to each other, whereas the blue color blindness is a separate thing, because both the genes, the L cone forming gene and the M cone forming gene, is present on X chromosome, but the S cone forming gene, that is opsinin 1 SW, is present on a different chromosome, that is seventh number of chromosome. So causes of color blindness, here if you see, then I have shown two different chromosomes. The first one is the seventh chromosome of human and the other one is the X chromosome. So here if you see in the seventh chromosome, on the long arm of the chromosome, here opsinin 1 SW is present, which is responsible for the formation of the S cones, which are going to give you vision of blue color, whereas the opsinin 1 LW and opsinin 1 MW gene are present in close proximity over the lower ends of X chromosome and on the long branch, or you can say long arm of X chromosome. So if there is any, even a slight mutation occurs in opsinin 1 LW, opsinin 1 MW, or opsinin 1 SW, then it may result in formation of color blindness. So as we know, opsinin 1 MW and opsinin 1 LW is present on X chromosome, then if there is any slight mutation in MW or LW gene, then it will result in the onset of your red and green color blindness. For general sense, the opsinin 1 MW having a multiple copies over the X chromosome and opsinin 1 LW have a single copy followed by the multiple copies of MW. Expression of both these genes, that is both opsinin 1 MW and opsinin 1 LW, are controlled by a common master switch, which is a LCR region, that is local control region. Okay, as I told you that both the genes are present in close proximity, so it may happen that they both result in recombination, and as we know, recombination will itself result in loss of genetic material from either one of the gene or from both of the genes. So if the recombination occur, or you can say if the loss of a gene occur, then it will result in color blindness, and depending upon which gene is getting mutated, the color blindness can be classified into 6 types. The first is tritanopia. ट्रिटेनोपिया particularly blue color blindness के लिए word use करा जाता है. Tritanopia is the absence of opsinin 1 SW gene, shorter wavelength gene, and it will result in the onset of blue yellow color vision deficiency. So the person having tritanopia will find difficult to distinguish different shades of blue as well as different shades of yellow. Now tritanopia, it is abnormal opsinin 1 SW gene. There will be no total absence, but there will be some you can say slight mutations that will result in formation of a mutated gene, opsinin 1 SW, and it will result in the formation of blue yellow color deficiency, but that will not be a you can say very severe case. It will be less effective. Now the other is deuteranopia, that is total absence of opsinin 1 MW gene, that as we know will result in the formation of red green color deficiency. But on the other hand, deuteranomaly, which will be the abnormal opsinin 1 MW gene, it will lead to less severe. It is true that the protein, the color sensitive, the light sensitive protein which is present in cone cells will be different, but in this case, but that protein will not result in total red green color blindness. Okay, there will be less severe case of red green color blindness during deuteranomaly. Now proteinopia, it is total absence, or you can say the protein, it is total absence of protein which is formed by opsinin 1 LW gene, because in front of opsinin 1 and LW there will be appearance of a stop codon that will totally cease the formation of the proteins. which will be formed by opsin 1 LW in normal condition. So it also result in red green color deficiency. And then protonomally, it is replacement of a normal opsin 1 LW gene with a hybrid gene, with a recombinant gene, or you can say. And it will also result in the formation of red green color vision deficiency, but in a very less severe form, not that much complex like in the protanopia, but a bit, you can say, less severe during protonomaly. So every student is facing difficulty to understand that if not red color, then what color. Everyone is saying like this, like if this person is color blind for the red color, then if not red, then what, particular uh, you can say color, or which type of vision they are getting. So I here picturize some you can say diagrams, or you can say some pictures that I get from the internet, that will help you in knowing that how a colorblind person will see the world. So the first picture is of a bunch of grapes, that purple color, for the normal eye person, but that having the, you can say, color blindness. For dichromat means having two color blindness along one another. So we'll see the bunch of grapes like this. And in this diagram particularly, the person having normal vision are seeing the particular your M&M candies, or you can say your gems, in such color. But the person who is deuteronomally, means the red green color blind person, will see this bunch like this. And the one who is having protonomaly, that is also red green color blindness, but due to the absence of the gene product of opsin 1 LW, so that person will particularly see the bunch of candies like this. And the person who is facing trigonomaly, that is blue yellow color blindness, will see the M&M candies like this. So now I think it will be very clear for all of you that how a person who is color blind is going to experience this colorful world, है ना. They may be not experiencing the colors like we are doing, with normal people are doing, but still they are getting colors, but that colors are different from us, because their cone cells are not working, you can say, correctly. So as we know that this is called as the Wavegear of human eye, that how much normal colors a person can see. But the person who is having green color blindness due to normally red green color blindness, for that particular person the Wavegear is like this. You know, that person is not having good differentiation in red color as well as in yellow or orange color. And the person who is facing protonomaly, and particularly the red color blindness, that is also not be able to distinguish between different colors of red, yellow and orange. And the person who is protonomaly, for them the particular whole spectrum is like different, but the main difference in this blue color region, as you can see, for them the blue is much less denser. For like us, for like this is very dark blue, but for them it is like sky blue, or you can say light blue color. Now inheritance means transmission from one generation to another. Like in very first slide I told you that both color blindness and the next we are going to discuss hemophilia is X-linked recessive disorder. So uh, as we know, the male carry only one X chromosome, so one mutated X chromosome is sufficient for the onset of disease, whereas female there is two chromosomes, and for the onset of disease color blindness the female has to get or inherit both the defected genes, है ना, affected genes. Otherwise there will be no color blindness condition in female. That is why male is more color blind, the number of color blind male is more than that of female. Female are very less affected. Okay, so by the inheritance pattern of X-linked recessive disorder, we can say that father will not be able to transmit the disease color blindness to their son, because father only transmit Y chromosome to son, not X chromosome. And the disease color blindness, the gene of this disease is present on X chromosome itself. Okay, but as we know that blue green color blindness is due to the seventh chromosome, which is autosome, so that particular disease is autosomal dominant. Okay, so here is a small inheritance chart. When the father is affected, that means it have the X chromosome which is having a defected gene. Okay, opsin in one gene जो particularly वो affected है, but the mother is completely normal. So in this case we have only carrier daughters and no son will be affected. Okay, that is a very characteristic feature of X-linked recessive disorders. But when mother is affected, then daughter is still carrier, but we will have 50% chance of a son being affected by a X-linked recessive disorder if the mother is affected, or you can say if the mother is carrier of the disease. Now, the next disease, that is hemophilia, also known as bleeding disorder, and a very fancy name to this disease also given, that is royal disease, due to its connection in the royal family tree. So, characteristics of hemophilia is slow blood clotting. Maybe if the hemophilia is not severe, then it may result in blood clotting, but that blood clotting will be very slow, so it will result in prolonged bleeding. And if the person, the patient, has undergone any surgery, then you can say is have any injury, then it will result in oozing out of blood after a small interval. There is a very special case known as spontaneous bleeding. That means the bleeding will occur without any severe damage, or you can say without any injury. It is more severe case of hemophilia. So, what will happen if there is spontaneous bleeding? Then it may occur that the bleeding may be inside the body, like in muscles, joints, brain, internal organs, and that will result in very serious complications. Type of hemophilia, there is you can say in broad sense there is only three type A, type B, and type C hemophilia. The type A hemophilia, known as classical hemophilia, it is just due to the deficiency of blood clotting factor 8, that I will going to discuss in some later slides, that what is blood clotting factors and what is particularly blood clotting factor 8. But if the person is lacking blood clotting factor 8, then it will result in formation of, or you can say onset of, a classical hemophilia, that is type A hemophilia. Now type B hemophilia, it is also known as Christmas disease. It is due to deficiency of blood clotting factor 9. Now the type C hemophilia, which is not a X linked disease, it is an autosomal disease, which is due to the absence of blood clotting factor 11. Okay, it is just due to the absence of blood clotting factor 11. Now the type B hemophilia, that is hemophilia B, sometime also known as hemophilia B laden, when what will happen when there will be mutation in the 5' promoter segment of 5 F9 gene. F9 gene is responsible for the formation of blood clotting factor 9, and if there is mutation in its promoter site, then it will result in you can say completely ceasing the translational process. So that is very severe form for the particular, you can say this particular condition of hemophilia B can be diagnosed in early ages, or at, you can say, just at birth. But after puberty, what happened? The androgens produced inside the body during puberty and after puberty will bind to the promoter side and can facilitate the formation of factor 9. So after puberty, the extent of this disease, that hemophilia B laden, decreases. So what are the causes? So the main cause that occur during hemophilia A is the mutation of at F8 gene, which is itself present on the X chromosome. So F8 gene result in the formation of protein, that is blood clotting protein 8, or you can say blood clotting factor 8. So if there is any mutation in F8 gene, then it will not result in the formation of the blood clotting factor, or you can say protein 8. So it will result in the formation of type A hemophilia. Till now, more than 1300 alteration of these genes has been identified. The most common mutations are like intron 22 inversion and intron 11 inversion. Okay, sometime a change in a single base pair, or deletion or insertion of multiple base pairs, can also cause particular mild hemophilia A conditions. Mutation in F9 gene, which is also present on the X chromosome, so it will also result in the formation of hemophilia B, that is particular Christmas disease, which is due to the deficiency of blood clotting factor 9, because we know that F9 gene is going to form the blood clotting factor 9. For the journal sec, the F8 gene is 186 kb in size and have 26 exons, and that means it is a very big gene. whereas the F9 gene is very small, only 34.7 kb in size, and have only 8 axonic regions. So you may be familiar with the coagulation pathway, that is particularly the blood clotting pathway भी कहा जा सकता है इसको. So there is two different pathways, the intrinsic pathway and the extrinsic pathway, that occur simultaneously and then merge into a common pathway. Okay, so I'm not going in detail of the coagulation pathways. I think everyone is familiar with coagulation pathway, but here are our special factors, blood clotting factors. The particular blood clotting factor 9 and particular blood clotting factor 8. So if any of these factors, or you can say proteins, will be absent or will be non-functional or hybrid, then they will block this intrinsic pathway. And if this pathway is blocked, then what will happen? The common pathway will be having very less effects, or you can say sometime it may also fail to work. So if this particular ninth factor is not working, then it will result in type B hemophilia, and if this eighth factor is not working, then it will result in type A hemophilia. So there is a diagrammatical view of the above diagram is of a normal person. So when this is a particular common blood vessel, any blood vessel. So as you can see, the blood vessel get damaged and there is a blood loss. But what will happen? As the blood loss occur, there will be result in the coagulation pathway, and which will result in fibrins, and fibrins will entrap platelets and then result in formation of platelet plug, and the blood clot will form. But for the hemophilic person, when the injury occur, then the fibrin will not form, or if the fibrin form, then it will be in very less amount. So the platelet plug that was going to form in a healthy person will not form in hemophilic person, so it will result in continuous loss of blood. Okay, so that's why the name bleeding disorder, no? The continuous loss of blood from any cut, or you can say injury. Inheritance, it is also sharing the common inheritance pattern with the color blindness, because it is also X-linked recessive disorder. So again, male carry only one X chromosome. So if only one mutated X chromosome is present, then it is sufficient for the onset of disease hemophilia. That's why male are having a very higher risk of hemophilia, but in female, two X chromosomes are present. So for a female to be affected with hemophilia, she must have to inherit two defective X chromosomes. Okay, that the both X chromosomes having defected F8 and F9 gene. So it is very rare. And as we know, for the X-linked recessive disorders, father never transmit this disorder to son, because father always give Y chromosome to son, and the gene responsible for this disease is present on the X chromosome, and mother can transmit this disease to son as well as daughter. So this is also two common, you can say, pathways are here. The first, you can say, the inheritance patterns are there. Here the mother is unaffected, but the father is affected, or you can say hemophilic. That means the X chromosome which is present inside the father genome is affected. So what will happen? No son will be affected, and both the, you can say, daughters are going to be carrier of this disease. And what will happen if a mother is affected, or you can say if mother is carrier and father is unaffected, then there will be 50% chance of a son being affected with this disease, and 50% of chance of a daughter being carrier of this disease. Okay, so this is all for color blindness and hemophilia. Thank you so much.